Facebook. Facebooking me. Facebooking.
Oh, who?
Jeff, you were talking about the streaming issue.
Oh, good.
Are you ready?
We're already
Should we
live.
get
You
going?
go whenever you're ready.
Okay.
You should turn your lights on.
Can everyone hear me? Cool. Alrighty, well, welcome to starting a QC program on a budget. Excited to be here. This is meant to be kind of an entry-level introduction to a QC program. If you're doing 10,000 barrels a year, this is probably going to be basic information for you, but I think this is designed for people who think that it's too hard to do a basic QC program or something that you have to hire a PhD to do. Like, these are tests that are designed for any brewer to do if they have the time or the specific resources to do them. So that's kind of our goal with this. Let's stand over here. So who are we? We're both from Bootleg Biology. We are a yeast company here in Nashville, Tennessee, established in 2013. I'm Jeff Mello, founder, chief yeast wrangler for Bootleg Biology. I do not have a background in microbiology. I was a poli-sci guy. Started off as a home brewer with a passion for wild yeast and microbes, and that kind of led me down the path of seeing how many cultures we can find in the world and how many are awesome for brewing, and trying to share those with the world. I'll let Isaac introduce himself.
Hey guys, I'm Isaac Brannon. Glad to be everyone's first SBC presentation this year. You can just go ahead and set the bar nice and low for everybody else. I first got my interest piqued in being a part of the craft beer community as a 19-year-old home brewer, trying to pick a college major. Closest I could get to homebrewing was microbiology. So after school, I met up with Jeff and helped get Bootleg Biology off the ground. So here I am.
Here's our contact information. Ever have any questions? If you want the presentation after we're done, email address. We're on social media, Twitter, Instagram, Facebook, or bootlegbiology.com. So just to get a little more detail on what Bootleg Biology is, we started the Local Yeast Project, which as far as I know, the first project to catalog and source wild yeast from every zip code in the entire US. We haven't done it yet. It's an ongoing pursuit, but our goal is to share with the world the wild yeast that's out there that's ready to make us beer. So since day one, part of that has been sharing the microbes, but also teaching people basic science skills. So we've talked at homebrew conferences. We have our website, which is, I think, pretty informative on how to wrangle yeast at home. And then also with this presentation to do entry-level explanations of wild yeast and wrangling and quality control. We created the Backyard Yeast Wrangling Toolkit, which was designed to help you capture and isolate yeast from your backyard so you can brew with it. And we also offer commercial-sized pitches of unique cultures, so Brett bacteria for souring, mixed cultures for funky sour beers, long-term aged beers, and then Arlingtonesis and Chardonnay were yeast that we sourced from outside in the wilderness in our backyards that are pure cultures that can make really tasty beers. And then we also do custom blends of all those things and any other brewer's yeast that's out there. We also provide services including IBU SRM testing, house culture banking, contamination check, cell count. So basically all the things that we're talking about we do on a regular basis, so we feel like it's something that we can share with you in a good way. Here's just an outline of what we're going to talk about. So before we get into specifics, I can't tell you how to start a program. I can't tell you what's the most important part of the program, but I think you have to ask yourself some big picture questions. You know, who's on the QC team? Well, the easy answer is everyone. It's everyone's job to be in charge of quality at your brewery. But you need a team lead to do these things. But you also need to find out, what are we testing? What do we want to learn? Don't just test the things that you've always been told to test. There's no point in wasting your time on those things unless it's going to give you something that's worthwhile. And then once you know what you want to test, you need to determine where are the thresholds for a negative or positive result on that test. A certain number for one brewery might be okay, and it could be negative for another one. Where's the information recorded? It's a no-brainer, but who's in charge of the information? Is it just written on a piece of paper and hung up on the brew house floor to get wet and destroyed at some point in time? Or is someone the keeper of the information and keeping a Bible of every beer, every single point that it's tested in the process? When are we testing? It's pretty obvious, but anytime you're touching that beer, anytime you're about to move that beer, anytime you're about to package that beer, it's ideal to test it. That may not work for you, but in an ideal scenario, that's where you'd want to test. And then yeast, day of harvest, before you pitch, anytime you're going to move that. And then just the big question is, why are we testing these things? No zombie testing. Don't test something if you're not going to do something with that information. If it's purely just for, that's interesting to know, there's no point. You're wasting your money, you're wasting your time, and if it's a negative result, then it just kind of beats up everyone for no reason. The ASBC has created an awesome guideline for all the equipment and testing you'd ever want to do, and it's excellent because it breaks it down by how many barrels you put out. Just give you a sense of, something for New Belgium is very different for someone who's just starting up their first 5-barrel brewery. But I think a question to ask yourself is, what is your budget? Well, what is the cost of dumping your largest batch of beer? If it's $200, then maybe you don't need to put too much money into it. If it's $2,000, that's a very different equation, because if you have to start dumping 3 batches of beer a week because you have a significant contamination program, that's when you can start considering certain equipment or hiring someone to handle all these things. And an important thing is, who can you trust to handle delicate tests? These are all doable that we're going to talk about, but you need to have someone who's attentive to detail, not everyone is. And then also, what is the cost of doing it yourself versus sending it out for testing? Some, I think, are better for sending out, some are better for doing in-house, but that's really up to you. Some of the equipment that's listed here, we'll go into a little more detail in the next slide, but it's like anything. You can find a used version of it on eBay. There's other sites that are meant more for government and universities, Surplus, DovBid, LabX. If you know the microbiologist at your local university, they can give you an inside track on where to get that equipment. A lot of universities constantly are cycling through really good quality equipment that they just don't use anymore for budgetary reasons or something else. So contact those people and talk to the suppliers of equipment specifically. They will come out and do demos. If that equipment is very pricey, they will try very hard to get your business, and so they'll come out and show you how to use a salometer or how to use a DO meter. These are, I would say, entry level for starting a QC program. You're on a budget, you can get all of these things for under $1,000, maybe $1,500. You don't have to get all of these things, but these are all, in the end, fairly inexpensive for starting up your own QC lab. So I'm going to hand it off to Isaac, and he's going to talk about how to do cell counting and viability checks.
All right, cell counting and viability. This is stuff that everyone knows we should be doing. Reasons being is, obviously, you want to hone in on having the right pitch rate for every batch of beer. Under pitching is going to lead to problems like under attenuation. It can stick you with pretty long lag times. This is problematic not just because it holds up tank space, but it's also going to give any slight contamination that you have in that tank a chance to start metabolizing wort before the yeast can start to produce alcohol and inhibit that. So you can get off flavors that way, but the yeasts are also going to leave some off flavors due to an underpitch as well. Overpitching, on the other hand, this is going to leave you with yeast that you're harvesting that is reducing in viability with each successive overpitch. So you can think of this as not enough beer for everybody or all the yeast cells at the party. Especially after multiple generations, you can end up overpitching yourself into a scenario where you're harvesting your yeast at less than 80% viability. which obviously isn't something that you'd want to repitch. Each time you finish brewing a batch of beer, you can double check your pitch rate by counting the yeast cells at time of filling your tank. So once all of your wort and your yeast is pitched in the tank, you can go ahead and grab a sample right out the sample port and do just a fill count and double check that you're in the right ballpark. All right, so how are you going to do these cell counts? I'm sure that if anyone had an extra $10,000 to spend on something at their brewery, first thing you'd want to buy is a fancy cell counter. These are super automated, very accurate, easy to use, really quick. They're worth every penny of that $10,000 that you have to spend on that cell counter. The downside, obviously, is the cost. In addition to the fact that with each different yeast strain that you're counting with an automated device like this, you'll have to adjust the size of the cells that it's looking to count, depending on the size of that culture or how it picks apart clumps of cells. Once you get that done, though, you can save that profile for each strain. So if you have 2 or 3 house strains, you set it to different settings for each house strain. So it makes it quicker once you get it done the first time. Inexpensive method. This is going to be what most of us are doing for the rest of our lives until we have that $10,000. This is with a hemocytometer. You'll also need cover slides specific to a hemocytometer. They need to be larger than your everyday cover slide, so make sure you're getting the type that are for a hemocytometer. You'll also need your viability stains, test tubes for your dilutions, and a micropipetter and pipette tips. I should mention that this is, even though this doesn't cost $10,000, this can be every bit as accurate as long as you know what you're doing with it. It's relatively easy to perform. It's also easy to perform inaccurately, so it does take a little bit of experience and skill. The stains you'll be using, we've got 2 different viability stains listed here. They both stain dead cells blue. You're probably mostly familiar with methylene blue. This is super easy to use. For that reason, it's kind of the most commonly mentioned for brewers to use as their first viability stain. You just drop a few drops in your final dilution, and then it stains your cells. The problem with it is that it can give both false positives and negatives depending on certain scenarios. So for that reason, we use trypan blue. It's definitely more accurate. A downside to it is that it can be toxic both to your own cells, so you'll need to wear gloves when handling it, and to yeast cells. So as soon as you stain your cells, it can start to kill off cells just over time, so you'll need to hurry up and get your count done once it's stained. So I'm going to try to take you kind of chronologically through the steps of performing an accurate cell count. Obviously, the first thing is going to be collecting your sample. Your yeast sample that you're counting, that slurry, needs to be a mirrored reflective density of the slurry that's in your brink. So you need to have your yeast slurry in your brink fully homogenized before you take your sample. The best time to do that is going to be right when you harvest your yeast. However, if your brink is going to sit in a cold box for a few days, it's definitely best to take another sample right before you pitch it and get another count. But be sure to take that sample once your brink is mixed up and homogenized again. At that point too, you're going to want to decarbonate or degas your sample. You're likely to have a bunch of dissolved CO2 in that sample, and if you try to pipette a carbonated yeast slurry sample, those CO2 bubbles are going to come out of solution in your pipette tip and start pushing clumps of yeast out the bottom of the pipette before you're able to put it into your next dilution. So if you can imagine how many millions of cells are in each little drop of yeast, and then extrapolate that to the cells that are in your brink, you're going to end up with a wildly inaccurate count if you're trying to pipette a carbonated yeast slurry sample. So best way to do that is either using an oversized collection container that you can shake up and kind of burp to remove the gas, or if you have to, you can pour your sample into kind of a clean and dry bowl and whisk it with a fork, and then just pipette from that. So once you've got a sample, you're ready to start diluting it. You're going to want to use a micropipetter, kind of like this one seen here. You can set that to a very specific pipetting volume. It's going to be 1 milliliter. These are preferable to the little bulbed dilution tubes or transfer pipettes, because every time you use a transfer pipette, you're going to be slightly off of that 1 milliliter mark, and that's going to throw your counts all over the place. Makes it difficult to really hone in on your proper collection and pitching techniques and all that. So firstly, you're going to pipe that 1 milliliter from your yeast slurry into a 9 milliliters of water. Shake that up really well, make sure it's nice and homogenized, and then pipe out another 1 milliliter from that into 9 milliliters of water. And then you've got a 1 to 100 dilution, which is going to be what you're going to use most often. In this case, these dilution numbers are for trypan blue. That particular stain needs to be staining at a higher, you need a higher volume of stain than you would with methylene blue. So you'll add 1 milliliter of trypan blue to your 1 to 100 dilution, which is going to dilute that further to 1 to 110. If you're working with a particularly clumpy yeast strain, English strains like 007 and 002 from White Labs, rather than diluting with water, you can use 0.5% sulfuric acid, which causes the cells to deflocculate, makes them much easier to both dilute properly and to get an accurate count on. A lot of sources will recommend EDTA for this. If you do want to do that route, you'll need to adjust the pH back down to a neutral 7.0 with sodium hydroxide, which is just more stuff to buy and more math to do. And sulfuric acid works just as well. So once you've got a dilution that is properly diluted and stained, you're ready to load your hemocytometer. Again, another thing that can be done easily incorrectly, it's kind of a delicate procedure. Unlike putting a sample on a slide and putting a cover slip over that just to look at what's in your sample, you'll need to put your cover slip on the hemocytometer first, and then as you see kind of this pipette tip is loading the sample underneath the cover slip, right where the cover slip meets the hemocytometer slide. There's a little opening right there, and it'll pull the sample in through the pipette tip. So if you look back at this picture, that is an accurately loaded hemocytometer. There's a moat right between the two counting chambers and on either side of the counting chamber. And it's really easy to see when your blue sample overflows from the counting chamber into any of those three moats. Then you've overfilled the hemocytometer. You need to toss the cover slip, rinse it off, and dry it with a Kim wipe, and try to load again. So once you've got your properly loaded hemocytometer, what are you even looking at? This entire 3x3 square is going to be the counting chamber, and then this 5x5 square in the center is where you're actually going to count. So the long count method is going to be counting that entire 5x5 square, so counting 25 squares. That's what you'll need to do if your cells are not evenly dispersed throughout that chamber. In the case that they are, it's equally as accurate to count the 4 corners and the one in the center. It's going to save you a lot of time. The math changes just a little bit, but it's just as accurate. There are a whole host of just rules to keep in mind as you're counting the cells in these chambers. They're listed in every brewery text about counting cells, so I won't go too deeply into them, but they're here for your reference. But I do have a quick example. So in the very top right, you see a cell that's stained blue. That's the color that a dead cell will look with trypan blue. Just beneath it, there's a daughter cell that's under 50% the size of the mother cell. So that would be too small of a daughter cell to count. Beneath that even further, there's a daughter cell that's over 50% the size. So in this case, you would count both those cells. And out on the left-hand side, there's a cell within that left-hand border. So you would not count the cell on the left-hand border, but if that were in this right-hand border, then you would count that. You can't really see these border lines very well in this picture. It's a little bit easier in the hemocytometer. But again, all of these rules, it's best just to keep in mind that consistency is the most important part of doing this. So if you're not the only person in your brewery doing cell counts, have these little rules just printed up and tacked right next to the microscope, and make sure that your whole QC team is on the same page as far as what to count and what not to count. Lastly, my absolute favorite part of cell counting, if anyone could have a favorite part of cell counting, is that there's an app that makes it a lot easier. So this is an app for an iPhone. There should be Android equivalents. This one is called HemoCyTap. As you're going through counting your cells, it keeps track of both the live and dead cells. And then at the very end, all you have to put in is your dilution, which in this case is 1 to 110, and your total volume of slurry that you're counting. But do keep in mind that when you're doing these cell counts, what you're essentially counting is cells per milliliter. So your entire count is only going to be as accurate as that initial volume that you put in. So if you have a yeast brink that you think feels relatively full, so you just put in 15 gallons, but truthfully there's only 13 gallons in there, your total cell number right here is going to be pretty wildly over the actual number of cells that you have in your brink. It is possible to do counts based on weight. For that you need a pretty big scale that you can actually weigh your yeast brink on. These can kind of be thrown off by hot matter and trub that are in there. And then in that case you would also need to do your dilutions based on weight using an analytical balance. So in all honesty, dilutions based on volume are a lot easier.
One more thing about HemoCyTap is great is you can email the results as a CSV. So if there is a point person on your QC team who's keeping track of all these things, it's easy just to keep a running spreadsheet of all the counts you've had for each strain. All right, contamination checks. How many people have done some kind of contamination check in a brewery before? How many people would want to do one if they could? If they thought they could. Okay. I assume everyone would want to know what's in their beer. And that's what the goal is, to know what's in your beer. You know what the hops are and the malt are, but what microbe besides what you pitch are in there. And then make an informed decision about what's in there. It's not always the end of the world if there's something besides 001 in your beer, but sometimes it is. But goals are prevent contaminated beer from being released, ensure your packaged beer is going to be shelf stable. It might be on a gas station shelf for 3 months at 90 degrees. That could be very bad. You can make a decision on how you're going to package it. You could be in a situation where the beer is actually pretty good. You don't taste any off flavors. You might have problems down the road, so you just keg it, and you know for the most part the supply chain is going to be cold, so you're not going to have any issues with microbes throwing off flavors or exploding cans. And then the big one is just preventing future issues. You can't take away the microbes that are contaminating a beer, but you can prevent that yeast from being repitched into another beer. You can adjust your CIP process. You can kind of having a moment of clarity where you go, we need to make a change. There's a serious issue here. There's a lot of methods for finding out what microbes are in your beer. Like Isaac talked about, there's the expensive options, there's the inexpensive ones. The expensive ones are very accurate. It uses PCR technology that's actually looking at the DNA that's floating in your beer. There's commercial suppliers out there that make them. They're highly accurate. You can know within a few hours if you have a contaminant. The con is very expensive. They don't necessarily tell you if the microbes in there are viable, meaning alive, and they don't necessarily test for every single microbe that could be in your beer, just the most well-known ones, maybe like bread or lacto or PBO. There are other things out there that are less expensive. There's Pica Fast Orange, which I think is relatively new. It's a pH indicator broth, so you put your sample in there, and if you have lacto and the pH drops, it'll change color. It's very simple. Anyone can do that test. Less work than making your own agar plates that we're going to get into. More expensive than making your own agar plates. And it has the same turnaround time, i.e. incubation time. You need to let the microbes grow to find out if they're in there, as opposed to PCR, which you can find out same day. We're going to talk about old school planning, which I would consider the gold standard of knowing what's in your beer. It's really cheap. You can enumerate. You can find out the exact number of bacteria or wild yeast that are in your beer, and that gives you a really informed decision on what to do with it. And then showing that they're actually viable, that they're in the beer and they're alive and they could wreak havoc at some point. The con is that it takes time to make your own agar plates. You could buy them online. They're pre-made agar plates that are available through lab supply companies. Those are so pricey, but it's easy to make them yourself. But another con is still there's an incubation time. You need to be able to plan ahead 3 to 7 days to find out the results. So there's a lot of media out there. These are the three that we would recommend the most if you're just starting up. WLN is great. It's primarily for yeast. There's some bacteria that can grow on it. It's differential in that different cultures will look differently. So visually you can tell that there's different types of yeast growing. MRS is specifically designed for lactic acid bacteria like Lactobacillus, Pediococcus. They grow really well on this media. You can add cyclohexamide, and cyclohexamide prevents the growth of almost all yeast cultures. And so that way you would know that whatever's growing on there is definitely bacteria. One small asterisk to that is that Brett does have a tendency to grow in the presence of cyclohexamide. WLD is a version of WLN. You can add cyclohexamide to a plate for yeast, and then you know if it's growing on there that it's not brewer's yeast. HLP is a great starter media. It's a lot pricier than WLN and MRS are, but it has the ability to recreate an anaerobic chamber. It has oxygen scavenging compounds in it. It's a lot easier to make in that you don't necessarily need an autoclave to sterilize the growth media, but it's going to be a little bit more expensive. So what do you do with these plates? Well, I think there's 3 easy methods of using them that'll give you good results. And you can use any combination of those agar media to do these tests. You can streak. Streaking is the most simple thing you can do as a microbiologist. You take a lab, which is a piece of metal, you flame it to sterilize it, you dunk it in whatever you want to sample, and then you just run it across the top of the agar plate. If it grows, there you go. Usually it's going to pick up high numbers of cells. So if you have 1 or 2 cells in your entire sample, probably not going to show up. So there are other methods for dealing with those kind of situations. You have a spread plate. So a spread plate is pipetting a very small sample, 0.2 milliliters. Isaac was talking about a micropipetter. You're going to need something that's very accurate to do that amount. And you just squirt it on top of an agar plate, and then you spread it around. We call it a hockey stick. It's cell spreaders. It's a piece of metal that you sterilize and spread it around on the top of the plate. You get an even distribution of cells. Pour plates are great for kind of recreating an anaerobic situation. You can see which cultures will grow better in oxygen, some that will grow better with a small amount of oxygen. So essentially you pre-make the agar, put it in a test tube. You can sit on the shelf if you autoclave it. And then when you're ready, you squirt 1 ml of your sample of beer into a plate that doesn't have any agar in it. Then you warm up the agar. Agar is solid at room temperature. At 110, 120, it's liquid. So warm it up just enough. You don't want it too hot, otherwise it'll kill the microbes that you pour it into. But once it's 110-ish, dump that into your sample, squish it around, and then there you go. You're going to have a plate that gives you a lot of good information. We'll give you some examples here very soon. So what is the point of doing these checks? Well, we need to figure out what's in the beer. Well, how do you know what's in the beer? We need to differentiate them. You can't use a microscope to identify the species of a microbe. That's not something you can really do. You need to do sequencing to do exact species, but that's okay. Really, genus is all you need to find out to make good decisions. You can guess that based on morphology, and morphology is just what does the microbe look like. You can look at microscope, cultures under a microscope, and you can generally tell a Brett culture because it has certain hallmarks. Lacto and Pedio have very specific hallmarks to their morphology that are easy to spot under a microscope. You also have ways of differentiating too. We talked about cyclohexamide prevents yeast from growing, so that's a good way of saying, okay, if it grows in the presence of cyclohexamide, it's not yeast. WLN, the first media we talked about, has bromocresol green in it, which is a pH indicator. So those plates that start off are blue. As the pH drops, they'll start changing to green or to yellow, so that you know that if it gets entirely yellow, you have an acid producer, a significant acid producer, and brewer's yeast isn't going to do that. The pH will drop a little bit because if it's yeast on a plate, it's going to produce ethanol, and ethanol will drop the pH. Certain hallmarks on WLN of cultures growing is, for the most part, and these are not the rules, these are just general guidelines, is brewer's yeast on WLN tends to be just dark green. That can change over time, but for the most part, it's dark green. Wild sac phenolic brewer's yeast strains like Saison tend to be white, and Brett cultures tend to have Very slow growth patterns. They'll go from different colors, yellow, white to green, and they'll have sometimes rings. They'll kind of look like flying saucers on an auger plate. Here's some examples. These are all WLN plates, streaked at the same time. On the left is a phenolic sac strain. It's pretty brightly white. On the top right, green, brewer's yeast. It's probably something like 001. And then Bruxellensis. As you can see, the kind of growth pattern there, it's going from, on the bottom right, whitish to yellowish to all the way to the very end, it's green. And so that could be happening over a period of days. Here's an example of spread plates. You can see a ton of cells on that plate. You really can't see much. There's so many cells. This was a very densely populated sample full of 001. But you can tell there's some abnormalities there. There's some big white dots. You don't even need to know exactly what that is, but you know that it's a yeast culture, and it's very likely a phenolic yeast culture based on the color and the size of the cell. And to the right is the pour plate. We're actually looking at the bottom of the plate, whereas the other one was the top. You can see there's dark green dots. On the flip side, it's going to be white. So you can see that that cell grows much better in the presence of oxygen, and on the bottom part where there is no oxygen, it's hardly growing at all. There are 77 contaminant wild yeast cultures in that plate, and that was just a beer sample. Other tests you can do, I'm not going to go into too much detail, but this is helping you to differentiate. Once you know you have bacteria, what kind of bacteria is it? Okay. There's a lot of bacteria that's not going to be in the world if it's in your beer. Obviously you don't want any, but if you have lacto and PDO, those are more likely to be beer spoilers. These are tests that will help you differentiate which ones are lacto and PDO versus other cultures. Problem areas, we all know these. We know cleanliness. Breweries are focused on cleanliness. Everything is about cleaning and sanitation. But there are certain problem areas that you're just going to more likely find microbes coming into your beer. A certain beer that's low IBU, low ABV for high final gravity is going to be the highest likelihood to be contaminated because it doesn't have the high IBUs to inhibit growth, it doesn't have the ABV to inhibit growth, or it has a lot of residual sugars that are just a snack and waiting for a microbe to jump into. Things like gaskets, yeast rings, spent grain, your ports, open doors, human beings coming in and out of your brew house and touching things. You are surrounded by microbes, so it helps to be paranoid in these situations. So, you know that there's a problem in your brewery. What do you do about it? Well, a good place to start is your CIP process. You ask a dozen brewers, you'll get a dozen different answers on what the CIP process should be. So, how do you verify that the CIP process is working? There's actually very helpful devices out there, the ATP limitometers, multiple manufacturers that make these. It's a way of testing for the presence of the elements of life. So it'll give you a number that estimates how much life essentially is in your tank. There's different types of swabs. Liquid swabs are great for testing your final rinse to ensure that you actually did a good, thorough job. There's no residual malt or yeast in your fermenter. Dry swab, you can do your ports, your gaskets, things that are hard to visually see, essentially dry areas. So that helps you figure out if you have a cleaning problem. But then, most importantly, to do any of these tests, you got to know how to sample correctly. There's a lot of ways to sample and get an accurate way of testing what's in your beer, but you want to make sure you're not contaminating your sample by microbes that might be on the outside of your sampling port, but also not introducing microbes into your beer by taking a sample to see if there are microbes into your beer. And that happens. So it's really simple, you know, spraying it, swabbing it, remove any dried beer that might be in the sample port. Alcohol, flame, you don't have to cook the end of the sample port, you just have to remove the ethanol. And most importantly, wear gloves. You want to be sanitary anytime you're taking a sample. There's a lot of neglected tests. I feel like every brewery does all these tests, or to some extent most of them, and it's really easy when we're understaffed, we're super busy, we're working late to do a gravity check and not do it accurately. How many people only use a hydrometer for testing gravity in the brewery? Yeah, it's okay. But do you have the time, are you taking the time to check to make sure your hydrometer is temperature corrected? Are you changing the reading based on what the actual measured temperature of your sample was when you took it? Those are easy to do. Everyone knows them. A lot of people know them from their homebrew days. There are far more complicated machinery out there, as the DMAs. Anton Par makes a ton of density meters that are very expensive. There's a machine that's marketed to homebrewers that's very accurate. It's called the Easy Dens, and you need just, was it 1 or 2 mils of sample to test the gravity, and it's designed to hold that information and store it so you can check it later. Things like pH, everyone should be doing. Sensory, are you drinking your own beer? This is a test. Enjoying your beer is one thing, but objectively tasting your beer. Are you drinking and tasting the finished product every single time? Because you're the first line of defense on catching a problem in your brewery before 3 months down the line when it goes to a consumer. I'm just going to touch on this lightly. I can't say it more fervently that please don't put your yeast in corny kegs. There are just a lot of issues with sanitation, and you can pick up contaminants that way. There are other things on the market that are designed for holding yeast that have sanitary fittings, and they're a lot easier to clean. They're a little pricey, but in the end, you're going to save a lot more money by using yeast in that storage capacity. I don't need to tell you to keep it cold to release the CO2 pressure, but these are the things you've got to think about. And especially like Isaac talked about, anytime you're taking a cell count with your yeast brink, make sure it's a homogeneous sample, that you're not just throwing off your counts by taking something that's either too much yeast or not enough yeast. So I think the takeaway is, okay, what do I need to do? Well, I can't tell you that. You need to think about how often do you want to test something? What exactly do you want to test? What are your priorities? Do you have problems that you want to correct? Or are you paranoid about certain problems because I just told you there's a bunch of microbes in your brewery? You need to make that decision, but start small. You don't have to test every single thing that's possible. The ASPC guidelines are great for making you paranoid, but they don't necessarily, are what you need as a startup brewery, or even you're only doing a few thousand barrels a year. So things like cell counts and contamination check, we hit those hard because I think anyone can do those and they're inexpensive. You can send them out. So if you think that you could do it, but you don't have someone you trust to do it, you don't have the time to do these counts, because it could take you an hour to do them, or a contamination check. Not that expensive to send it out, but that's up to you. You can make that choice. Things like IBU and SRM are great for testing to know if your beer is shelf stable, knowing if you have enough IBUs to prevent contamination, knowing that your marketing materials are correct, but also just knowing the consistency of your hop utilization. But is that important for you? If you're struggling just to get your beer packaged into market, maybe IBU isn't important because your beer is drank 5 hours after it's packaged. But you can send that out, or you can do it yourself. But I would recommend sending it out if you could. ABV, I don't know many people that actually test ABV on a regular basis, at least on the small brewery scale. You have a certain amount of tolerance by the TTB. It's not much. I can guarantee you if you have an over-attenuative yeast strain that's contaminated your beer, you're very likely going to be out of compliance by the TTB. The alkalizer is super accurate. It's very expensive. Only the largest breweries, I would recommend having those, but it's inexpensive to send it out. Send out all your flagship beers. If you brew this beer 20 times, 100 times, 1,000 times, and you've never checked the ABV, other than trying to make a calculation based on density, I think you will be very much surprised on what the actual alcohol content in your beer is. Lots of other testing you can do, diacetyl, DMS. I think a lot of people think that they need to test these things all the time, but it's usually something that could be caught from your in-house sensory panel. So if you're drinking your beer every time, or even better, your in-house sensory panel is people who don't brew the beer. A disinterested person is going to give you a lot more critical feedback than a person whose job might be on the line. You need someone who's going to give you brutal honesty about these things. But you can send them out, $90, $100 to test. I think White Labs does like the big QC day where they do a wide panel of testing. Those are super helpful. It doesn't have to be done every time, but it could be a good way to open your eyes. So, key takeaways. You need to decide before you start the program how much you're willing to spend, how much time you're willing to give to this, and if these tests are worth it for you. It's not just something you need to do because you're told to, but you think you can do something with that information. Cell counts, contaminant checks, you can do those. You just need to find the time. The cost really isn't an issue. Don't neglect your common test. take the time to do them right. Verify your CIP is being done correctly. You know, have a protocol that everyone uses, and then verify it with something like an ATP luminometer. Don't contaminate your own beer sample. Sample your beer the correct way. Use ethanol, flame it. And then, in the end, you're deciding what's important to you for quality control. And I think everyone is here because this is something that's important to them. We're here at a conference talking and learning about beer because it's important to them. Rely on fellow brewers, people in your community who are working at labs, people who are microbiologists, people at your local university. Everyone wants to help everyone make better beer, and I think you can totally do a lot of these tests yourself. Special thanks to the ASBC, who does fantastic work. If you have someone who is a QC lead for your brewery, make sure they're a member. They're always doing studies about beer. There's methods of analysis, which really anything you could ever test in your brewery, they have a method for it, and they have multiple ways of testing for it. Brewer Association obviously has helped pull us all together, and then the Southern Brewers Conference for making sure we have a venue for us all to get together and talk about how great we are as brewers. Don't be a stranger. We're located just a few blocks south of here on 3rd Avenue, and we're having a get-together this evening, so there's a lot of after parties, but you're all welcome to come by anytime between 6 and 9. We can show you what we do, pour you some samples of homebrew, about show off our cultures, and just talk yeast and geek out. So, thank you. I don't know how we doing on time.
Yeah, you got 4 or 5 minutes at least.
Okay.
Just repeat the questions so people hear.
Okay. Anyone have any questions?
You talked a little bit about luminometer. Is there a specific brand that you would suggest using?
I think Hygienia is one we've used. I think Charm or something is another one. They're all relatively in the same price range, and I think they all pretty much do the same thing. They just may have different bells and whistles. Sorry, I forgot to repeat that. Is that you asked if there's a brand of ATP luminometer to use? So I don't have a specific recommendation other than we've used Hygienia and it's worked really well. I think they are very competitive price-wise, so you can just find the dealer or the salesperson who will give you the best deal and go with them.
I have a question.
Yep.
I might have missed it, so I apologize. You said terpene blue was better than methylene blue, but I don't know if you said exactly why it was better.
I might have skipped it. Trypan blue it's got a greater accuracy when staining live or dead cells. Methylene blue has a tendency to throw out both false positives and negatives in different scenarios. And it's almost as easy to work with.
And I think someone had talked about this with the ASBC, but someone did a test of every single viability stain they could find, and in the end, their takeaway was Trypan blue was the most accurate. All right. Cool. Well, thank you all for coming and listening to us yammer on, and let me know if you want to email you the presentation.